Thanks for the nice introduction, and welcome to the particle size session. The first topic today will be on advanced microscopy methods. But before we start, here is a message from the Office of Pharmaceutical Quality. A quality product of any kind consistently meets the expectations of the user. Drugs are no different. Patients expect safe and effective medicine with every dose they take. Pharmaceutical quality is assuring every dose is safe and effective, free of contamination and defect. It is what gives patients confidence in their next dose of medicine. Here is today's learning objectives. From this presentation, you will learn what are the current challenges in characterizing particle size distribution for complex drug products. After this presentation, I hope you can name 2 advanced Raman microscope methods. And from a case study, I hope you can get a basic idea what's the general consideration for advanced microscope method development and validation. Particle size distribution of API in the drug product is a critical attribute in evaluating quality and effectiveness of complex drug products. It is also an important attribute for bioequivalence evaluation of generic drugs. What's to note here, we are talking about particle size of APIs, not excipients. However, in reality, a lot of drug products include both API and excipient particles in the formulation. And in some cases, the drug product may include more than one API, and the API may have more than one polymorphic form. Traditional particle sizing techniques such as sieving, laser diffraction, and microscopy, they treat all the particles, no matter API or excipient, as the same. They cannot distinguish particles with different chemical identities. Therefore, those techniques cannot provide the measurement we need. In recent years, development of advanced microscope methods provides a solution for this problem. Most of advanced microscope methods use Raman and microscopy combination technology. If you are not familiar with Raman spectroscopy, Raman provides information on molecular vibrations and crystal structures. Raman spectrum are fingerprints of molecules. Each molecule has its own unique Raman spectrum. API and excipient particles show significantly different Raman profiles, therefore they can be easily distinguished. By combining Raman with microscope, it giving microscope additional chemical identification power. However, the disadvantage is the overall measurements are relatively slow, and sometimes it is a very time-consuming process. One example of advanced microscope is cryo-SEM Raman. Cryo-SEM Raman is a combination of 3 techniques: cryofixation, SEM, and Raman into one unit. The highlight is its advanced sampling technique, cryofixation. It is a rapid freezing technique able to preserve samples in their native states. This is particularly useful for evaluating pharmaceutical emulsions and biological products. In addition, unlike regular microscope, which measures particles in micron size range, SEM can measure particles in nanoscale size range. Another example is morphologically directed Raman spectroscopy, short as MDRS. The highlight of MDRS is its advanced analytical methodology, which applies a morphology screening procedure to significantly reduce total measurement time. MDRS has been used as an alternative approach to the comparative clinical endpoint BE study. It first used as supporting evidence for the approval of the first generic mometasone furoate nasal suspension spray in 2016. And recently, it has been recommended in several FDA product-specific guidance. In this presentation, we will use MDRS as a case study for advanced microscope method development and validation. As mentioned before, Raman measurement is relative time-consuming, and what we interested are API particles only. We do not want to spend extra time to measure excipient particles. For a lot of drug products, API and excipient particles have different morphological features. MDRS utilizes a morphology screening as the first step to exclude as many excipient particles as possible before any Raman measurement. Only the remaining, a small portion of excipient particles will be further excluded by Raman confirmation. Overall, due to the majority of excipient particles were excluded from Raman measurement, the total MDRS measurement time is largely reduced. Some commonly used morphological parameters are listed in these slides. Several to highlight. First is the aspect ratio, which is the particle width over the particle length. Elongation is the reverse of aspect ratio. Convexity and solidity measures the surface roughness of a particle. And the intensity mean describes how transparent or opaque is a particle. A typical MDRS protocol includes 4 steps. They are sample preparation, morphology screening, Raman confirmation, and finally, report API PSD result. The most important step of all is the second step, morphology screening. It is important to apply appropriate morphology filters. A loose morphology filter will cause biased API PSD results. Therefore, method development is the key to a successful MDRS method. MDRS method development and validation focus on 4 areas. Other than sample preparation, the most critical step is to gain a complete understanding of morphology and chemical ID of particles in the drug product. After we gain a good understanding of the system, morphology filter selection will be a relatively comfortable process. And finally, we need to determine the minimum number of particles need to be measured. It's a balance of the result accuracy and the total measurement time. Here we use the nasal spray suspension as a case study. Every good analytical method starts with a good sample preparation. Our goal here is to keep the sample in its native state, with minimum sample preparation involved. The reason is if the sample needs to be prepared in a diluted or dried condition, the process of dispersion, ultrasonication, evaporation, or freezing may cause changes in particle stability. In that case, validation will be required to ensure the particles are still measured in their native state. We were able to have a sample preparation without any dilution and drying. An example was shown here. First, we gently shake the nasal spray bottle and prime the pump, then deliver 2 actuations into a sample well. After that, we transfer 5 microliter sample onto a microscope slide, cover it using a cover slip, and apply petroleum jelly to seal the edge to prevent sample evaporation. Finally, we allow time for the particle to settle. There are 2 key considerations during sample preparation. The first is the volume of the sample. The volume of the sample should be minimized and optimized for producing a reproducible thin layer. First, it will avoid possible overlapping particles or touching particles. Second, it will provide a better focus on all particles in the field of view. Second consideration is on the time of settlement. When the sample was first prepared, the particles will still moving around. It takes some time to settle. The settling time will depend on the viscosity of the formulation. The high viscosity formulation normally takes less time to settle. After sample preparation, it's time to gain a complete understanding of the particles in the drug product. We will use a large training set, include as many particles as possible. Here I will just throw a number, at least 10,000 particles. We will perform morphology and Raman measurements on each of the particles in the training set. Later, we will classify all the particles based on Raman into two classes, API and excipient, for further morphology analysis. Before we move on, I want to mention again about touching particles, or agglomerates, aggregates. Some of the applications choose to exclude them from further analysis, but it may not be a good practice all the time. If more than 5% of particles are touching particles, it indicates a poor sample preparation. You may need to go back to revisit your sample preparation procedure. If more than 5% of particles are agglomerates or aggregates, it may indicate a potential stability issue. Those particles need to be investigated as a separate class other than API and excipient. All the particles in the training set can be classified into API and excipient groups based on Raman. Raman classification itself is a relatively straightforward process. The only thing I want to mention here is the limit of detection for MDRS. The limit could come from both the sample and the instrument. The limit of detection for the current version of the MDRS instrument can be as good as 1 micron. For the drug product studied in this case study, after Raman separation, the visual image shows us the excipient particles are generally longer and more transparent than API particles. The visual observation can be further confirmed by comparing the morphology parameters of the API and excipient particle groups. The morphology parameter plots shown here indicates that the best separation of the API and excipient particles can be provided by aspect ratio or intensity mean, which indicate that those two parameters could be the best filter candidates for morphology screening. Another information we learned from those plots are that none of their morphology parameters can provide a complete separation between API and excipient particles. That's why morphology can be only used as a screening tool. After nailing down aspect ratio and the intensity mean as a filter selection, we need to choose proper cutoff values for them. By using the morphology filters, we want to exclude as many excipient particles as possible. But at the same time, we also need to realize the filter application will inevitably discard some API particles due to overlapping morphologies. In our study, we set a goal to keep at least 85% API particles after applying morphology filters. The ultimate goal for a good morphology filter selection is to ensure applying morphology filters will not change the PSD results. Since morphology filter selection is the most critical step in MDRS method, it needs to be well validated. We can reanalyze the training set by applying morphology filters with different cutoff values and evaluate the PSD result before and after filter applications. The criteria here is to make sure D10, D50, and D90 have less than 5% difference after filter application. If the criteria cannot be met, the filter needs to be tightened to include more API particles. The last but not least step is to determine minimum number of particles to be counted. The goal here is to ensure enough number of particles are measured so the PSD result is statistically meaningful and can represent the actual product. The minimum number of particles should be determined by evaluating the accuracy and the repeatability of PSD measurements. We need to ensure measuring more particles will not result in significant changes or improvement in PSD results. On the other hand, the PSD measurements and the results should be repeatable. A simple method to do this is to compare PSD results from counting different number of API particles. The criteria for evaluating the accuracy of the measurement is to check if the D10, D50, and D90 have less than 5% difference if more particles were counted. The criteria for evaluating repeatability is to check the relative standard deviation, RSD, of the D10, D50, and D90 from 5 replicates should be less than 5%. The minimum number of particles depend on the particle size distribution profile. The number required could be significantly increased if you have a wider particle size distribution profile or have a multi-model distribution. Here comes the summary. Advanced Raman microscope methods are capable to perform automated imaging, high-resolution particle morphology measurement, chemical identification, and polymorphic form identification. Some of the advanced systems can even perform in-depth investigation of API excipient interaction in complex drug structures. However, advanced Raman microscope is a relatively complex analytical method, and they are also emerging technology. Therefore, extra effort is required in order to develop robust methods, and there are always more room to improve in both hardware and analytical methodology. Additional summary is given for MDRS to highlight considerations needed to be taken during method development and validation. During sample preparation, we want to prepare sample in its native state as a thin layer for better focus and dispersion. For particle morphology analysis, we need to watch for possible agglomerate aggregates for potential stability issue. And then we need to realize the limit of detection for Raman microscope. Morphology filter selection is the most important step in an MDRS method. We need to make sure to avoid biased filter selection and to ensure PSD results do not change before and after filter selection. Last but not least is the minimum number of particles. We need to ensure enough number of particles were measured so our PSD result is statistically meaningful and can represent the actual drug product. At the end of this presentation, we have a poll question. Which of the following morphology parameters are used to describe particle shape? A. Aspect ratio. B. Solidity. C. Convexity. D. All of the above. The answer is D. All of the above. Finally, I want to thank for the collaboration from my FDA colleagues and the GDUFA research funding from OGD. and I will see you again in the Q&A session. Thanks for your attendance.